defense. Okay, so Miranda, let me tell you a little bit about Miranda. She's a master's candidate in the Environmental and Sustainability Studies program. She has been the recipient of the prestigious Slocum Lens Foundation grant, as well as a grant from the college's Center for Partnerships to Improve Education. She presented some of her research at the Second World Conference on Marine Mammals in Barcelona, Spain, which is the largest marine mammal conference to date. She was the second author on one paper in PLOS One and the first author on a paper recently accepted to GeoHealth, all before she graduated. And she's also this year's recipient of the Outstanding Graduate Student Award for her program. So congratulations, Miranda. Originally a Michigander that attended the University of Toledo in Ohio, she majored in biochemistry, which made her perfectly suited for this project. She moved to Charleston sight unseen to pursue a master's degree that would be the perfect blend of science and policy. She loves the small town feel of Charleston and spends much of her free time on Folly Beach, but she does miss those Midwestern seasons. This was her first experience working with dolphins, and besides her interest in ecotoxicology, she loved learning about their social patterns. So before we get into Miranda's defense, I'd like to introduce her committee members. So we'll start with Dr. Randy Wells. He is the Vice President of Marine Mammal Conservation for the Chicago Zoological Society and the Director of Chicago Zoological Society's Sarasota Dolphin Research Program, which is the world's longest study of wild bottlenose dolphins. We also have Dr. Ed Wirth, the Chemical Contaminants Research Lead here in Charleston at the Hollings Marine Laboratory, which is operated through the National Ocean Service, a branch of the National Oceanic and Atmospheric Administration. On the phone, we have Dr. Brian Balmer, formerly of the National Marine Mammal Foundation, and calling in all the way from Montana because he is now the Environmental Contaminants Lead for the U.S. Fish and Wildlife Service's Montana Ecological Services Field Office. And we have Dr. Susan Lovelace, who's the Executive Director of South Carolina Sea Grant Consortium here in Charleston, located just across the street from the College of Charleston. And I'm Leslie Hart, Assistant Professor of Public Health here at the College of Charleston. So Miranda's going to be talking to us about the continuation of some pilot work that we did that demonstrated exposure to plasticizers in bottlenose dolphins. And like most research studies, that pilot project led to more questions than answers. And so Miranda wanted to tackle some of those questions, particularly regarding exposure vulnerability. So without further delay, I'm going to hand it over to Miranda and she'll tell you all about the work she's been doing for the past few years.
Okay. Oh. Okay. Well, Leslie, thank you so much for that introduction. That was very generous, and I really appreciate it. So, yep, excuse me. My thesis was the characterization of phthalate exposure in common bottlenose dolphins from Sarasota, Florida during 2010 to 2019. So just a quick general outline for this presentation. I'll give a brief introduction on phthalates, what they are, why we care about them, how we can study them. I'll go over the importance of marine sentinels, the Sarasota Dolphin Research Program, and then the overall objectives for this study. So these objectives are broken up into 3 chapters that comprise my thesis. The first was a demographic assessment of mono-2-ethylhexylphthalate and monoethyl phthalate concentrations. And as Leslie had just mentioned, I'm pleased to announce that this chapter of my thesis has actually been accepted for publication in the journal GeoHealth. The second objective was a temporal and spatial evaluation of MEHP detection. And finally, the third objective was a correlational analysis of MEHP and hormones. Then we'll bring some context back into this project, talk about some current policy efforts, the overall conclusions from the study, and then the next steps moving forward.
Hey, Miranda, I hate to interrupt,
yeah.
But we can see your notes.
Ah, that's so embarrassing. Okay.
No, you're totally fine. We can currently see them. So if you want to adjust that.
Do you know how I would do that?
There should be like do you have a double monitor right now?
I do not. Well, when we had done the practice runs, you couldn't see my notes before.
Correct.
So
There is something you can toggle that'll make it go back and forth. A student did it the other day. I don't know what that is.
can you
though.
see it right now?
We can.
It was just working fine and then went back to the previous way.
Whoops.
Is the split screen the one that's in the middle? That one? What is?
Miranda, what if you, yeah.
We just need to, apologies, everyone. I promise this did work before. Okay, I'm gonna stop sharing for a second while I can. Try that again.
I do think there's a way though that you can see your notes and not have to see them.
What if you just try like presenting as you normally did on your computer and then share your screen?
Okay.
Oh, that's a good one.
Well, I have to do it because then if I present it, it takes up the whole screen, so I can't get back to the Zoom.
So what I've done in the past, or what I did yesterday, is
Yep.
you share your screen and then have your
Yep.
notes up, but only share a portion of your screen. So you do this and then put your notes up, and then you will go back to reshare and it'll say share a portion, and you can adjust it to only see that
Oh,
screen. Does that make sense?
I think so.
So keep sharing it.
Well, like share the screen rather than the application?
Correct.
Or what do you mean?
So yeah, you would just share your screen instead of just the application. So
Okay.
go back to this, and then
Yep.
you'll go to slideshow, and then go to presenter view. Now put your little clicker up at the top so it'll give you a range where you can reshare your screen. It'll come down a little green button. Do you see it?
Yep.
Click that and it'll say share a portion of your screen, I believe. And then you will toggle that to only see the same screen, but where we can only see your PowerPoint of that screen. Does that make sense?
Yes, but I'm not seeing anything that just says share a portion. What are you seeing right now? Do you see like just the slide, or do you see the notes too?
I see the notes as well.
Oh, okay. Oh, wait, how about this? Wait, that's the wrong thing, sorry. Oh my gosh, okay. Okay, how about now? Is it still notes?
No, it's good. Can you see your
Okay,
notes?
for some reason I had 3 PowerPoint tabs open, and one of them was presenter view, and one of them was like my actual presentation, and then okay.
Okay.
Still
Well,
can't see notes?
nope, you are good to go.
Oh my gosh, okay, I'm so sorry, everyone. Anyways,
All right.
that
Sorry
hiccup.
to interrupt you. Now you're good to go.
Thank you for the help, Lucy. I'm glad that you said something. Okay. So as I mentioned, here's the brief introduction of phthalates. So what are they? Phthalates are a class of chemical ester. They're commonly referred to as the everywhere chemical because of their wide-ranging use. Predominantly, they're used as a plasticizer with PVC to soften and extend the durability of the plastic. However, they can also be found in personal care products, insecticides, pesticides, cleaning solution, and a variety of other commercial goods. You can see some of those examples in this table here. Phthalates are not chemically bound to the products they're added to, so they can easily leach out into surrounding surfaces. As humans, we face exposure through inhalation, ingestion, dermal absorption, and intravenous infusion. Once they're in our bodies, they're metabolized really rapidly through 2 phases, where we then excrete their metabolites through our urine and feces. So this table, this first column lists some of the more commonly used phthalates, and then the second column is going to be those associated metabolites that we're actually excreting. Exposure is important because human and laboratory studies have linked phthalate exposure with serious reproductive and developmental effects. This is due to interference with normal activity of hormones and their receptors. So this diagram just shows an example of how endocrine disruption can function. This example specifically shows the endocrine disruptor mimicking the function of the normal hormone with its receptor, but disruptors can also bind to receptors affecting synthesis and transport of hormones throughout the body. Reported health effects associated with phthalate exposure include physical reproductive malformations, neurodevelopmental deficiencies, thyroid tumors, and organ toxicity. And some laboratory studies have even shown evidence that health effects can be long-term even after exposure ceases. So if we look up here, this is the overall structure of what a phthalate is. There's 2 classes that phthalates can be categorized as. We have a high molecular weight phthalate and a low molecular weight phthalate, and that's determined based off the length of carbon chains linked here at these R groups. So the type of phthalate is also really important in determining resultant health effects, because that can determine what those effects are. Phthalate metabolites have even been detected in marine animals, which is a really concerning indication of marine pollution. We can measure contamination through sentinel or indicator species. These are animals that are sensitive to environmental changes and reflect the overall health of their environment. So a really classic example is going to be a canary in a coal mine. Marine mammal sentinels are particularly important because findings from these studies can be used to inform on human health. And phthalate metabolites have been detected in a variety of marine species, including harbor porpoises, fin whales, sea turtles, and the bottlenose dolphin. So, quick spoiler, one theme we'll see over the course of this study is the significance of MEHP contamination. So as Leslie had mentioned, we published a study last year. This one wasn't actually included in the thesis, but was conducted using the same samples, where we compared dolphin urinary MEHP contamination detection with human detection, and we were able to demonstrate that dolphins actually have higher MEHP concentrations than humans. The human data came from the National Health and Nutrition Examination Survey, or NHANES, which is conducted by the Centers for Disease Control and Prevention. So this was a really important finding because we know from literature, from previous studies, that the exposure that humans are facing is linked with health effects. So if dolphins are facing a higher exposure, as indicated by this higher detection, what does that mean for their health? Marine environments can be exposed to phthalates through a variety of routes, including sewage and wastewater discharge, as you can see here in this diagram, plastic manufacturing, plastic production, industrial discharge, agricultural runoff, and plastic pollution. So plastic in particular is really concerning because there's evidence of trophic transfer, which means that marine predators at the top of their food chain may be exposed to higher amounts of plastic particles via the prey they ingest. As plastics degrade, they release phthalates, so high amount of phthalate exposure may be related to high amounts of microplastic exposure. It's also important to note that the routes of exposure and type of phthalate that's the animals are being exposed to is likely different dolphins to humans. As humans, we're actively exposed to these chemicals through the products that we choose to use every day, whereas marine animals are exposed passively by means of environmental pollution. So a pilot study was conducted in 2018, and this was the first to detect phthalate metabolite concentrations in bottlenose dolphins sampled from Sarasota Bay. This study was enabled by the Sarasota Bay Dolphin Research Program, which is conducting the world's longest running study of a wild dolphin population. Efforts from SDRP have supported research to understand health, population dynamics, social structure, behavior, and reproductive success of the animals. And the Sarasota Bay population is made up of about 160 members that show high site fidelity, which means that the dolphins in the Sarasota Bay area tend to only be in the Sarasota Bay area. This high site fidelity has enabled long-term research efforts that utilize a variety of methods. The ones most relevant to this study were the photo identification surveys and the capture release health assessments. So overall, this thesis project was really exploratory. We didn't quite know what to expect because it's such a new area of research. We leaned on well-tested methods using 2 approaches to direct our objectives. The first was a One Health approach, and this is using the idea that findings from human health studies can be used to direct our objectives and our hypotheses. The other approach was a sentinel species study. So we knew from the pilot study that these animals were facing some level of exposure. So through long-term study of dolphins, we were able to examine whether one group may be more vulnerable to exposure, geographic location of increased vulnerability, and correlations with health indices. So as I had mentioned before, this project had 3 specific goals, and they were all based around expanding that pilot study. The first objective was to evaluate demographic vulnerability to exposure. The pilot study was unable to compare metabolite detection demographically due to a small sample size. Other pollutants, such as PCBs, will accumulate in an individual over its entire lifespan, resulting in clear detection patterns directly related to the age of an individual. Females also have a mechanism to clear their chemical body burdens through gestation and lactation, while males do not. So this also results in clear sex-related detection patterns. Phthalates are not expected to bioaccumulate, so detection patterns, particularly those in marine mammals, were unknown. Our second objective was to evaluate detection across spatial and temporal scales to see whether exposure may have been influenced by when the individual is sampled or by where it tends to spend its time. And finally, our third objective was to evaluate relationships between phthalate metabolite concentrations and serum hormone levels. We know from previous human and laboratory studies that phthalate metabolites have been linked with altered levels of a series of hormones. So we sought to understand if this could be observed in the dolphins as well. So just for some quick orientation for those of you not so familiar with the Sarasota area, Sarasota is on the Gulf side of Florida, located directly south of Tampa Bay. The dolphins can range anywhere in the Sarasota Bay area. So as a result, photo identification efforts and health assessments also range throughout the bay. So getting into our methods, the first method I'll talk about is the phthalate metabolite screening. It's not specific to any of the 3 objectives, but it rather applies to all of them. And this is how we actually detected and quantified phthalate metabolite concentrations in the dolphins. Human studies have demonstrated urine is a sensitive matrix to detect metabolites. So that's what we went for with this survey or with this study. So I think the question I'm most often asked when I describe my project to people is, how do you get dolphin urine? To collect urine, simple enough, we first have to catch the dolphins. So to do so, this occurs during SDRP's capture release health assessments, where a seine net is dropped in the water, we'll encircle the dolphins, and then once they're in the net, they're lifted aboard a research vessel where, as you can see from this picture, trained veterinarians can insert a catheter to facilitate the flow of urine into falcon tubes, where they can be aliquoted, stored, and shipped. I was fortunate enough to be able to attend the health assessment conducted in 2019, where we collected some of our urine samples. And then due to the extensive study the nature of extensive study being conducted in Sarasota, I also had access to archived urine samples, which I was very grateful for. And this really increased our sample size. So we actually ended up with samples obtained over a 10-year period. So once we had the samples in the lab, the first step was actually to separate the phthalate metabolites from everything else in the urine. So to do so, we use solid phase extraction, which is what's demonstrated in this diagram. We'll load the urine samples into this column, wash out everything we don't want, and then elute the metabolites afterwards. From here, we apply the samples to a high-performance liquid chromatography machine coupled to a triple quadruple mass spectrometer. Essentially, that's just a super complicated way of saying that we're separating the individual phthalate metabolites and then identifying and quantifying them via their masses. Each sample is run with a series of carbon labeled standards to identify each metabolite in the sample. And then we also run field and lab blanks to ensure contamination doesn't interfere with any quantification. So moving on to our actual objectives, the first one I'll go over is the demographic evaluation. So, and this is going to be consistent throughout the whole study. We did have a number of individuals with non-detectable phthalate metabolite concentrations. So to mitigate this and utilize our sample size to its fullest extent, we use we calculate our descriptive statistics using methods for centered data. We first wanted to compare based on sex and age class of the dolphins to understand whether one demography may be more susceptible to contamination than another. Sex comparisons, really straightforward, male versus female, but age comparisons required a little bit more attention. We decided on 2 age classes. The first one was adults and the second one was calves/juveniles. Age classes were determined using sexual maturity, and this was based on a number of factors for each dolphin, including the age, calving history, pregnancy diagnosis, and sex hormone concentrations. Demographic comparisons were done in 2 ways. First, we wanted to compare the proportion of detect, so the proportion of dolphins with detectable MEHP concentrations compared to the number of those without detectable MEHP or with detectable phthalate concentrations. The second comparison we wanted to understand was whether one demography may be contaminated more or have a higher magnitude of phthalate metabolite detection. So the first table on the left shows data regarding our dolphins. Overall, we had 69 urine samples from 51 individual dolphins. Some of our dolphins were sampled more than once, so to prevent any bias, we only used the most recently obtained sample. We had slightly more females than males and twice as many adults as juveniles. And then over here is our phthalate information. We screened for 8 total phthalate metabolites in the urine. The 2 most commonly detected metabolites were MEP and MEHP. Because of sample size limitations, these were the only metabolites that we used for this analysis moving forward. So overall, the results from this objective were pretty unsurprising. So this first table on the left is showing the comparison of the proportion of detects. As you can see, based on these p-values, there were no significant differences in the number of dolphins with detectable MEP or MEHP concentrations. And then this figure on the right shows the geometric mean comparisons between demographic groups among dolphins with detectable metabolite concentrations. So when I said this was unsurprising, that's really getting back to this idea that metabolism is really rapid and the chemicals are not expected to accumulate in the individual. Though it's not significant, I will note that, as you can see here, females did have higher geometric mean MEHP concentrations than males. So the overall takeaways from this objective, our findings were pretty consistent with previous marine studies. DEHP and DEP are produced in massive quantities every year, so it's not surprising that we found signs of exposure. We found non-differential MEHP and MEP detection, which indicated vulnerability to exposure does not vary demographically, so everyone is at risk. We did find that females may have increased vulnerability as they had higher MEHP concentrations than males, but this was not statistically significant. We were potentially underpowered to make those determinations, so further research with an increased sample size is warranted. So the next step was to evaluate where the dolphins tend to spend their time to see if there is any potential spatial influences in detection, as well as examine when we obtain the samples to see if sampling year also had any impact. For this objective, we only focus on MEHP for this analysis because it was the most commonly detected metabolite, and also this was due to our previous findings comparing dolphin and human exposure. We initially wanted to compare temporally by year, but our sample size required us to group sampling years to have statistical power. This resulted in some natural divisions between the years that we were able to use to determine our cohorts for comparison. So I'll get into that a little bit more in the next slide. But the comparisons were done using a Kruskal-Wallis test, and then we conducted pairwise comparisons with the Duns test. So this plot on the left shows the geometric mean concentrations for all individuals sampled in each year. It became pretty clear early on that there was something going on with 14 and 15. Not only did they have, or did they appear to have significantly higher geometric mean MEHP concentrations than all other sampling years, but they were also the only years where 100% of the sampled dolphins had detectable MEHP concentrations. So this was the basis for which we grouped our years. So 2014 and 2015 were grouped together. All years before and all years after were grouped. So our cohort 1 was comprised of dolphins sampled during 2010 to 2013. Cohort 2 was 2014 to 15. And then cohort 3 was dolphins sampled from 2016 to 2019. I will note as well that everyone was assigned to a single cohort. So I'd mentioned previously that we did have a number of non-detects, but we excluded some of the non-detects. So there was no, or excuse me, not non-detects, repeated samplings. So there's no overlap between groupings. Everyone was statistically analyzed only once. So as we suspected, cohort 2, which again was 14 and 15, had significantly higher MEHP concentrations among detects than both cohorts 1 and 3, as you can see by these p-values in the table. Similarly, the frequency of MEHP detection was significantly different for cohort 2. And actually, interestingly here, all 3 cohorts were significantly different in the frequency of detection, and dolphins from cohort 2 were more likely to have detectable MEHP concentrations. Overall, it was really interesting to find such substantial differences between the cohorts, both in terms of the magnitude and the frequency of MEHP detection. The first potential explanation we explored was laboratory or sampling inconsistencies that could have contaminated our samples. However, one thing that I would like to make abundantly clear is that it probably wasn't my fault. All of our methodologies, including sampling and quantification methods, were consistent throughout the entire sampling period. Additionally, all of our urine samples were run in batches and they were randomly sorted. So it's really improbable that only 2014 and 2015 concentrations would be artificially elevated and no other years would also be. Human studies have shown diurnal fluctuations and urinary concentrations that could have played a role in that elevated detection, but we were unable to analyze if this could be occurring in dolphins. Human studies have also shown that these concentrations only fluctuate about 2 nanograms per mil, so it's really unlikely that this fluctuation would have counted for our findings. In addition to those 2 explanations, we also were thinking about some potential acute exposure that could have accounted for our findings. We did look for evidence. So some of the things that we investigated were the things that I talked about before. So what we're showing in this diagram: sewage spills, agricultural runoff, and some other additional potential sources of an acute phthalate exposure include hurricane activity, nurdle dumps, and sewage spills. And those previously have been shown to be related to high amounts of phthalate contamination, particularly in marine environments, but we were not able to find any relevant explanations for our findings, unfortunately. So with those temporal findings in mind, we'll move on to the spatial analysis. The spatial analysis utilized records from monthly photo identification surveys conducted throughout the Sarasota Bay by SDRP. Using these surveys, information is recorded regarding geographic location of the individual, body condition and behavior, as well as a photo of the dorsal fin, as you can see in this slide. Dorsal fins are sensitive to tearing and scarring, so we can regard them similarly to human fingerprints, where it's unlikely that any 2 dolphins are going to have the exact same dorsal fin markings. So you can see here in this example, some pretty significant differences between the dorsal fins that would allow us to identify the individuals. Photo ID records were then paired to the urine samples collected during health assessments for analysis. Human studies have shown that single spot urine samples are indicative of average exposure over a 3 to 6 month period, but we ended up using photographic records for 1 full year prior to sampling to increase our sighting sample sizes. So these photo identification data were used in GIS to generate kernel density estimates, which is how we determine spatial use. KDE is a way to estimate probability. So in this case, it's the probability of a dolphin using space in a particular area. KDE assumes the higher density of sighting records are more important to the individual, so we're able to consider areas that are preferentially used over others. We looked at spatial use in terms of their utilization distributions, where 50% UD describes the core area of use, or where the individual is most likely to be, and the 95% describes the total area of potential use. So if you look at this figure that I made, super talented, I know, this 95% UD represents the total area that the dolphin could be, and then this 50% is this area of preferential use. We conducted 2 analyses. We first wanted to compare dolphins with detectable MEHP concentrations with non-detect dolphins to see if there could be any spatial explanation for why an individual might have detectable MEHP or not. We also wanted to further explore our temporal findings, so we compared spatial use across the same time periods that we used for the temporal analysis. So these maps are the detect versus non-detect comparison. So this figure on the left shows the 50% UD. Again, this is the core area of use, while the figure of the right shows the 95% UD, or the total area. For both figures, the lightest shade signifies the area that was determined to be exclusive to dolphins with non-detectable MEHP concentrations. The darker shade for both figures signifies the area exclusive to dolphins with detectable MEHP concentrations. And then this medium shade is the area of overlap between the 2 groups. There's quite a bit of evidence from literature that's relating MEHP detection with urban land use. So I included land use in in my figures. So this green area represents agricultural and farming land use. The blue is high density residencies, and then the black is industrial or commercial land use. To understand these findings, I looked at the study area in two sections. The first is this northern portion of Sarasota Bay that's indicated by this red box. In this area, we saw a pretty even mix of detect and non-detect dolphins, and this was indicated by that larger area of overlap. As you can see from the land use, this northern area is mostly residential and agricultural. There's some commercial land use, but this area also receives a lot of fresh inputs from the Gulf of Mexico, which is located right here, that could flush out any contamination from that area. So those could be potential explanations for why we found detect and non-detect dolphins in that area. The southern portion was defined as the area spanning from New Pass to Venice Inlet. And in this area, we found detects almost exclusively. This area has more industrial and commercial land use, which could contribute more significant amounts of phthalate pollution. This area is also far more closed off to fresh inputs as compared to the northern part of the bay. So any contaminants that make its way into this area is likely to stay in the area. Further, and you can't tell from the map, but there are a lot of freshwater streams and creeks that could be carrying inland pollutants and dumping them into the bay, just further increasing that amount of phthalate pollution. So overall, results from this first analysis suggest that there could be a potential relationship between urban land use and MEHP detection. Examining trends over the temporal groupings only utilized the core UD. This decision was made because mammalian metabolism occurs quickly, so the 50% UD maximized the likelihood that phthalate exposure occurred in the determined spatial area of use. Again, similar as before, the lighter area signifies, or the lighter shade signifies area exclusive to non-detect. The darker shade signifies area exclusive to dolphins with detectable MEHP concentrations. And then this medium shade signifies the areas of overlap. So I'll talk just in terms of cohort 1 and 2. Cohort 2 was a little bit of cohort 1 and 3. 2 was a little bit of an anomaly. So between cohorts 1 and 3, area in the north seemed to be pretty similar in terms of its exposure risk. Again, we see this larger area of overlap in the northern section of the bay. Interestingly, detection status seemed to shift between cohort 1 and cohort 3. So in cohort 1, hopefully you can see, but there's a little piece of this core UD that belonged to dolphins with non-detectable MEHP concentrations. And then in cohort 3, we see that shift, and this area is almost exclusively used by dolphins with detectable MEHP concentrations. And then going on to cohort 2, it was really interesting that all of the dolphins sampled in this grouping had detectable MEHP concentrations because they were sampled in the same area where prior to and directly following, there was an even mix of detects and non-detects. So this again points to this idea that maybe there was some sort of acute exposure event occurring, but we still don't know what that could have been. So finally, our third objective was to evaluate potential relationships between phthalates and hormones. Similar to the previous analysis, we were limited by sample size, so we focused on MEHP for this objective. Hormone levels are measured in blood. So following capture during SDRP health assessments, trained veterinarians use a butterfly catheter to draw blood from the ventral fluke, as you can see here in this image. Samples were then stored and shipped to Cornell University for hormone quantification. Included in this analysis were cortisol and the 3 thyroid hormones, T3, T4, and FT4, as well as aldosterone, progesterone, and testosterone. There was an assay change that occurred in 2014 that required us to analyze testosterone in 2 time periods. So the sampling years 2010 to 2014 were quantified using a radioimmunoassay, and then sampling years 25 to 2019 were quantified using a chemiluminescence immunoassay. Unfortunately, these assays were not comparable, which is why we had to keep the testosterone analyses separate. Hormone levels can be influenced based on the sex or age of the individual, so we compared the hormones demographically to characterize that variability. Once we understood which hormones may demonstrate demographic variance, we used a generalized linear model to determine our stratification strategy for these correlations. And with those strategies in mind, we were able to use the Akritas-Steele-Sen line to compute Kendall's Tau correlational coefficient and the p-value. As I had mentioned before, due to the number of dolphins with non-detectable MEHP concentrations, we use methods for censored data, and this is where this statistical test came from. So this table shows our sex-based comparisons. We found only cortisol and progesterone varied significantly based on sex. And this is our age class-based comparisons. We found, again, that cortisol and progesterone varied by age class, as well as T4 and FT4. So these are the results from the GLM, which again was conducted to determine how to appropriately stratify given our previous evidence of demographic variation in hormone concentration. Our model found age class to be an important term for cortisol and T4, and we also found sex and age class to be important terms for progesterone and FT4. T3 and aldosterone were the only hormones that did not require any demographic stratification methods, and testosterone was not included in the GLM. Aside from one adult female and one juvenile male, we only had adult male samples, so those were the ones that we used for the analysis. So these figures that I'm about to show are only going to be the significant correlations. So we found a significant positive relationship between adult female FT4 and MEHP. So this first figure is the untransformed data, and the second one is going to be a log transformed or a natural log transformed MEHP. We found a significant positive relationship between adult male FT4 and MEHP. And we also found a significant relationship between adult male progesterone and MEHP. However, this relationship is likely due to the influence of these two outliers rather than being a true correlation. So interestingly, our findings were actually not that consistent with previous studies. For the most part, evidence from literature has shown MEHP to be inversely linked with FT4, but we found the positive association. Our study was, however, most similar with findings from studies conducted in female children. Discrepancies between studies could be the result of metabolic differences. Previous studies have shown phthalate metabolism to vary between terrestrial mammals, so it's reasonable to assume that variability extends to marine mammals as well. It's important to consider contaminant metabolism because in general, metabolism should detoxify xenobiotic compounds. However, with the case of DEHP, what we see is MEHP is the more bioactive compound. Disruption to FT4 levels can have significant impacts on overall health. So this diagram just illustrates overall thyroid gland activity. So the thyroid gland produces T4 and a little bit of T3. However, the majority of T3 production comes actually as the conversion of T4 to T3. And then through a negative feedback mechanism, T3 regulates thyroid activity. So we can see how variations in T4 concentrations can be really important. There are many health implications related to FT4 disruption. In adults, these health effects have included dementia, mortality, frailty, bone deterioration, and increased bad cholesterol. Disruption can also have significant effects on a developing fetus. Fetuses are completely reliant on their mothers for T4 and they are unable to produce their own. So that means that they're really sensitive to changes in normal T4 concentrations. Increased fetal FT4 has been associated with reduced growth, low birth weight, and even fetal death. So overall, we were able to demonstrate that dolphin health may be impacted, or that we have this idea of thyroid homeostasis where normal thyroid function, hormone production is potentially being impacted because we saw that relationship with MEHP. Phthalates are man-made compounds, so the most effective way to prevent negative health effects is by mitigating exposure. Phthalate use is not highly regulated in the US. In fact, there's only one federal legislation regarding use, and that was a rule written by the Consumer Product Safety Commission, and all that says that this list of phthalates are not allowed to be used in children's toys or childcare articles in concentrations greater than 0.01%. Beyond federal law, some states have implemented their own mandates. So for instance, California has banned DBP and DEHP from use in personal care products. It's important to recognize that these regulations only apply to products made within the United States. As a country, we import billions of dollars worth of toys and goods from China, and these account for almost 40% of our total imports from this country. The products that we import are not required to follow any phthalate use legislation. So as a result, children still may be exposed to harmful levels. So the overall goals of this project all revolved around understanding phthalate metabolite detection in bottlenose dolphins. To do so, we analyzed a number of factors that have been previously shown to influence detection, including demographics, geographic location, and sample timing. Factors that did not appear to influence detection were sex and age class, and factors that did appear to influence detection were geographic location and sample timing. We found that FT4 was positively associated with MEHP in both adult females and adult males. This study was the first to characterize phthalate detection in the marine mammal community to this extent, but there's still a lot of knowledge gaps that need to be addressed. Like Leslie had previously mentioned, with these exploratory studies, often we have more questions following the study than what we first started out with. So findings from this study should be compared to studies of other dolphin communities, as well as other marine sentinels, to understand how our findings may or may not be extrapolated. Methods should also be further explored to detect marine mammal phthalate metabolites in matrices other than urine. Urine is a really sensitive and effective matrix, but it's not always available for all of the animals that we want to study, and it can limit the scope of research. More research also needs to be conducted regarding potential exposure sources, both for mitigation purposes as well as to understand resultant health effects, because health effects can vary based on the route of exposure, how much of the chemical you're being exposed to, and how long that exposure occurs. Finally, further research is warranted to identify whether the associations that we found with FT4 are causing negative health effects in dolphins. Our previous study demonstrated dolphin MEHP concentrations are actually higher than humans who are showing negative health effects, but we don't know yet how this will impact dolphin health overall. Okay, so a lot of technical difficulties at the beginning. Long and dense presentation, but if you'll just bear with me for a couple more moments, I have many people that I would really love the opportunity to thank who made all of this research possible. First and foremost, I would like to extend a huge thank you from the bottom of my heart to my lovely research advisor, Dr. Leslie Hart. None of this research would have been possible without her constant encouragement, enthusiasm, and support, and I am very, very grateful that she chose to take me on as a student all those few years ago. I would also like to thank my committee members, Dr. Randy Wells, Dr. Ed Wirth, Dr. Brian Balmer, and Dr. Susan Lovelace. I was really fortunate to work with people who were not only willing to guide me through this project, but really encouraged me to lean on them and their expertise, and for that, I'm really grateful. A huge thank you to the Sarasota Dolphin Research Program staff members for their dedication to bottlenose dolphin conservation that allowed me to even conduct this research. In particular, I'd like to thank Dr. Christina Toms, Jason Allen, and Aaron Barleycorn for their assistance throughout the project and for facilitating valuable fieldwork opportunities that I was so fortunate and so grateful to have been able to be a part of. I'd also like to thank Emily Pizarski for her mentorship and endless patience she had for me in the lab. Thank you to Amanda Morris, both for her assistance in the field as well as in the lab. Thank you to the EVSS department for their overall student support. In particular, thank you to Lucy Davis for humoring me with the hours she let me spend in her office muddling through my graduate degree. A big thank you to my parents, my siblings, my extended family, and all of my friends for just being a really solid support system. And then a big thank you to the organizations who financially supported this thesis. In particular, I'd like to thank the Department of Health and Human Performance for all of their financial contributions. That, you know, this goes without saying, we can't do research without being financially supported. So I'm very grateful to all of these organizations. And finally, thank you to everyone who bared with me throughout this presentation. Not everybody can get as excited over dolphin urine as I can, so I really appreciate the opportunity to speak about the work I've been doing. So as we mentioned a couple of times, if you want to learn any more about what the work we've been doing, we do have 2 papers that have been published. The first one is a comparison between phthalate concentrations in dolphins and humans. And then the second one was that demographic analysis from this thesis. So this first one was published in PLOS One, and then the demographic analysis was published in GeoHealth. So with that, I'm ready for questions, if anyone has them.
First, let's go ahead and give Miranda a round of applause via Zoom.
Thanks.
Terrific job. And we will go ahead and open that up to public questions. And again, you can go through the chat, or what I recommend is just speaking up if you have any direct questions.
Miranda, I'm happy to go first. This is Brian, since nobody else chimed in.
Sweet.
So awesome presentation. Your thesis was a pleasure to read over the past couple of days. Really phenomenal job. So start off with just kind of a question. Of course, it's going to be about spatial analyses. So the results of your different phthalate concentrations in relation to animal movements and spatial patterns of land use are pretty cool stuff.
Mhm.
Can you talk about any of the biases to using photo ID data for evaluating animal movements? And then similarly, what additional data could build upon your results to be collected in the future to better correlate phthalates, animal movements, and land use?
I'm sorry, what was the beginning of the first question? I missed it.
The biases of photo ID, was that the first part? Did you get that?
Yeah, I keep missing the first word. I'm sorry.
Biases.
The
So
bias.
is there any
Biases in
issues with
photo ID. Okay. Yeah, so some of the biases, especially for the kernel density method that we use, can be introduced just by sighting effort. So if you're only looking for dolphins in a certain area, then you're only going to find them in a certain area. So that was one of the really big benefits with working with SDRP, is their photo ID efforts span the entire length of the bay. In addition, that was also kind of one of the drivers to grouping our temporal years. The more data we were able to use, the less one individual could be biasing the data. And then your second question was, sorry.
Sorry. Yeah. So what next? Are there any other type of data sources that you can collect to build upon animal movements and relating that to land use and phthalates?
Sure, for sure. Yeah. So with the photo ID, I mean, you're limited just because you don't know where all of the animals are at any given time. So I think that some sort of like GPS tracking in addition to land use would be really cool, just to see specific movements over a longer period of time. I think it would also be interesting to compare. So we can regard, especially in terms of some of more polluted areas. So Sarasota tends to be a cleaner area than, say, other areas in the Gulf of Mexico where these big oil spills are occurring. So I think it'd be really interesting if we could compare what we find in Sarasota with some of these more heavily contaminated areas. I think it would also be interesting if we could compare them to like more rural or remote areas, just to see, you know, how we fit into this bigger puzzle, if that answers your question.
It does. That was great.
Okay.
Thank you.
Thank you.
I'll go next. Miranda, could you go back to your slide where you have the human body and the process of, oh, no,
Yes.
you passed it.
Oh, the
Go that
thyroid.
way. The thyroid one, yeah, sorry.
Yeah.
Okay, so you were able to participate in the health assessments in Sarasota Bay in 2019.
Mhm.
And you have demonstrated a possible link between MEHP and FT4. So what kind of data could we also use from the health assessment, given what you know about the samples that are collected, to try and look at these broader health impacts that are listed on this slide? Like, do you think there's any additional data to support your findings?
Well, one thing that we were unable to look at with this study was the effect of other contaminants. So, and that could really play a role in some of the health effects that we see manifesting, as well as the relationship we're seeing with the hormone and with the phthalate metabolite detection. So there could be, you know, combinations of however many contaminants in Sarasota Bay. I know there's been reports of PCBs, mercury, PBDEs, and so these all can have different effects either with each other, so they can act synergistically or antagonistically, increase the amount of health effects you're seeing, decrease the health effects. They can, you know, if one, let's say PCBs are decreasing your FT4 concentrations, but phthalates are increasing FT4 concentrations, and then you overall have like a net zero. So it depends. And so what I would really like to see moving forward is an overall contaminant study where we actually are able to compare multiple different types of contaminants and see how that affects health overall.
Sure. And just to follow up, so if we
Yeah.
wanted to look at some of these health impacts on this slide in dolphins, like what kind of data could, what specific things could we glean from the health assessments to try and answer those questions?
Well, okay, so with FT4 in particular, I mean, some of these, I mean, dementia, I don't know how you would measure that in dolphins, but birth weight and overall size seems to be related directly to FT4. So just measuring morphometrics and seeing how relationships, you know, over time or between individuals, and then relating that back to their MEHP detection. So overall, I would say morphometrics is probably a pretty good starting point, at least.
Great, thanks.
Okay, thanks.
Miranda, kind of going back to similar to what Brian was asking, I keep going back to the 2014, 2015 potential acute exposure. So can I ask you to kind of speculate maybe what other species we might want to look at if we wanted to try to tie down what may have caused that exposure?
If we're just looking at exposure, I mean, so the ideal thing about dolphins was eventually we hope to be able to compare them to humans because we are both mammalian species, top level predators. But if we're just looking for signs of exposure, and I'm not super familiar with all of the aquatic fauna that live in Sarasota Bay, but I would start out small, something like mussels or oysters, just to see if there is detection occurring. And those are easy that we can pop them in, take them out, and we can do, you know, a more finite temporal scale and see how exposure may be changing.
Good idea. Thanks.
Miranda, I have a question.
Okay.
So science is a very collaborative process, and our program is very interdisciplinary, and we talk about working with different organizations and how to partner with different organizations. And with this project in particular, you partnered with a lot of different organizations. Can you talk a little bit about that experience and how it overall increased the impact of your work? And let's say you had more time and you didn't want to spend 5 years in this program. Are there any other partners that you would have liked to bring to the table to increase that impact?
Ooh, that's a fun question. Well, first of all, I mean, I really enjoy collaborating with people. I love group projects. I love that aspect because everyone has different levels and areas of expertise, and you're never going to know everything about the things you think you know. And so that's where, you know, working with other people, you can kind of identify those areas, furthering your own knowledge. So just the collaborative nature in general was really valuable for me. And then had I the chance to work with other organizations, I mean, I think it would be really interesting if we could sort of, because I can do this research all day long and I can show you data after evidence that shows health is being affected, phthalates are out of control, this is crazy. But if there's no way to mitigate, and so I'm thinking in terms specifically of policy and of social science, I think that would be a really interesting component to see. So we have all of this information now, what do we do with it, and how do we, you know, make things better overall?
That piggybacks onto my next question.
Okay, I knew it was.
They're fun ones. How would you communicate these findings to make the general public interested in the overall?
Well, dolphins are kind of cool just in that they're a charismatic megafauna, and so people tend to care about dolphins more than they would like a mummy chug. So I think that's valuable in itself. So yeah, I guess I would lean on dolphins as that thing that people care about. I don't know. I mean, this is not so much my world, so I would have to again collaborate with other people to figure out the best way to communicate. But yeah. That doesn't really answer your question, but.
Miranda, this is Randy.
Yeah.
And I really like what you've done with the land use approach. I think it
Thank you.
Really brings out a lot of correlations. In order to do mitigation, you probably have to have pretty strong evidence that there's particular sources of these contaminants. And I was wondering how you might design a study that would get at identifying how these contaminants are getting into this ecosystem?
I mean, first, overall, it's going to take a long period of time, but where I would start, I think the things that most stood out to me in terms of exposure sources were plastic pollution. And then I think this was a comment that you had made in my thesis talking about inland streams and creeks that could be carrying contaminants from inland and dumping them into the bay. So if we can monitor those streams and creek and either identify them as an exposure source or say they're fine, that's not it, I think that would be huge, especially in this area down here where we don't have many fresh inputs. And so this, looking at the bay as it is, I would imagine this is a pretty sensitive area to chemical contamination. So that's where I would start. I would do a microplastics and I would look at these streams and creeks.
And so some kind of a sampling scheme for
Absolutely, yeah, like
water, for sediments, for
water monitoring, certainly. Sediment, definitely. I know phthalates tend to partition out of the water column, so sediment monitoring, and this could be getting back to Dr. Lovelace's question too of other organisms that we can study if we can sample some smaller species that live in or around those streams to correlate whatever we're finding in the sediment with what's actually being measured in the organisms. I think that would be really interesting.
Yeah, I think one of the areas to look at more closely is that Philippi Creek input down just inshore of Siesta Key, because it drains so much of the Sarasota area. And once, as you mentioned in your thesis, once Midnight Pass was shut down, the flushing of that area was greatly minimized. So I think there's good reason to put a lot of attention towards that in future work that you're doing.
Absolutely.
So Miranda, can you hear me?
Yep.
Okay. I'd like to follow up on this spatial, I don't want to call it an anomaly because we're not really sure, but you see this very different pattern in 2014 and 2015.
Yep.
You know, Susan has asked you about what other species you might look at. Randy's talked about how to monitor maybe a longer term assessment of what made this special, but what factors could you also look at right now historically to see if there are any other correlations that or relationships that might indicate that 2014, 2015 is very different? Do you have any thoughts on what you might be able to retrospectively kind of target data-wise?
Yeah. I would say probably water quality data would be a decent place to start. Again, phthalates aren't generally known to, they'll partition out of the water column, but I think that could kind of give some insights as to the overall health of the bay. I did look into rainfall data. I looked into salinity a little bit, any droughts, and nothing really seemed to be relevant to the timing of our sampling. If we could, if there was the data available for other contaminants that were detected in this area at that time, I think that could be a good indication, again, just overall health of the bay. Yeah.
Okay, and then a follow-up to that would really be, so you've said that phthalates partition out into sediments, I think in your last two answers, which is fine.
Yep.
Why do they not partition and bioaccumulate in the dolphin then?
So phthalates are really hydrophilic, so they won't, they're not stored in the fatty tissue cells, which means that they stay in solution, so then they can be transported through our liver. And in the liver, we have all of these enzymes that can metabolize. And so once they start being metabolized, they become even more soluble, because that's the whole point of mammalian metabolism, is we're trying to figure out ways mechanistically to get rid of these chemicals from our body. So more hydrophobic compounds such as PCBs, they'll bind right tight into the fatty tissues, and they're not available to be enzymatically metabolized in the liver the same way that phthalates are. And the more they're metabolized, the more soluble they are, and the easier they are to excrete from the body.
Okay, thank you.
Do we have any other questions from the general public? Okay. So with that, we are going to give Miranda one final round of applause via Zoom. And then we are going to, and then we're going to ask that our general public please leave, and our committee members please stay on.
Great job, Miranda.
Thanks, everybody.
Good job, Miranda.
Thank you.